Hello, this is Dr. Hooper, again presenting our webinars on environmental testing for mycotoxins and molds. This one is entitled EMMA, E-M-M-A, which stands for Environmental Mold and Mycotoxin Assay. What does it all mean? This is part 3 of 3 of these webinars. We're going to go back and review what we have. Webinar number 2 was Be Your Own Advocate for Environmental Assessment. It was part 1 of 3, Mycotoxins and Fungal Infestations in Indoor Environment. Then there was a second one, webinar series, which was held in March of 2019, titled Environmental Mold and Mycotoxin Testing, which was Introduction to Testing. And at the very bottom of this slide, you can click through to go to the YouTube presentation of this mycotoxin and mold webinar. So the summary of webinar 2B was historic availability for mold testing. We talked about the air testing and the tape lifts, etc. And what is bulk sampling. We also talked about spore traps and how does that relate to spore counts. We also talked about ERMI, definition of Hurts Me Too, and EMMA. Again, if you want to go to our learning centers, you can see our complete list of webinars that are present by clicking through to that. Today, the goals of webinar number 3 are we're going to explain why EMMA was conceived, and what are the components of EMMA, and what are the functions of the test in the product sheets, what are the explanations of the mycotoxins in EMMA, and then the mold/DNA in EMMA. We're going to talk about the introduction of a new concept that is being introduced today and will be introduced in publications in the manuscript we're preparing, and that is fungal load in the EMMA reports. Then we're going to ask about how do you order EMMA and what are the instructions for collection of samples for EMMA. So biggest question is, what does EMMA mean? How was it conceived? Well, the name EMMA is a girl's name of German origin, meaning universal. It's ranked number 1 on popularity charts in the naming of babies. This is weird, but it was my grandma's name. Really, that's true. It was. And it's an easy name to remember. Emma, get over here. Emma, let's do an Emma. So the real name is Environmental Mold and Mycotoxin Assay, which does not come right off your tongue, so you use the word Emma. How did I come up with that, and how did our group come up with that? Well, we reviewed all of the ERMI organisms, and we asked ourselves, what's the significance of all of these ERMI organisms when reviewing pathogenicity in human illness? There were too many organisms in ERMI, which caused confusion to the clinicians and the environmental inspectors as well. So, Real-Time Lab pick 10 of the most significant environmental organisms which can be pathogenic to human and animal patients. Now, this doesn't mean that this is all there is, but these are the most common ones, and as time goes on, we may be adding more. So why do we need EMMA? Well, the mold in a house isn't just a problem for people with allergies or asthma. Water leaks in homes provide an ideal environment for mold growth. We've talked about that. There are many mold species. Stachybotrys chartarum is one of the more dangerous ones than the most common indoor molds, such as Aspergillus and Penicillium. Mold exposure can cause immunosuppression and upper respiratory infections. Long-term exposure to these molds can cause acute to chronic illnesses. So the further background on the need for EMMA was really interesting, because as we did more and more prodding and studying this, we find that there's invasive fungal infections, especially invasive candidiasis and invasive aspergillosis, These remain the leading cause of morbidity and mortality in adult and pediatric hospitalized patients, whether they have leukemia or whether they're immune compromised or cancer or transplantation. The resultant problem that occurs with these patients are extended hospital stays. This causes an increased medical cost and very poor prognosis. The hospitalized patients with the highest risk for contacting invasive Candida, candidiasis, and aspergillosis are well written in the literature. So what are the components of EMMA and what do we test for? So there's DNA of the 10 most common human pathogens, which we talked about. There's 15 mycotoxins of the most harmful mycotoxins discussed in peer-reviewed literature. We've talked about aflatoxins. B1 is the most carcinogenic, but there's other ones, B2, G1, and G2. Ochratoxin A, macrocyclic trichothecenes, which are actually a group D trichothecene, are the most difficult ones to get rid of, and they are very, very pathogenic and or disease causing. And gliotoxin. Now, oftentimes you'll hear physicians, and such as myself, etc., talk about disease causing, but we want to not say that as much as disease associated. There's not been well documented literature to say that these cause a problem, but they are associated with these diseases and these presentations of symptoms. The EMMA test in the DNA testing is a quantitative polymerase chain reaction procedure, or qPCR. It detects 10 pathogenic fungal species in environmental dust specimens by looking at the DNA. It includes 10 assays that meet EPA standards, and these were developed and validated by Real-Time Lab. We are presently preparing a manuscript to publish all the validations of EMMA in an environmental peer-reviewed journal. This is a list of Aspergillus species tested in EMMA, and we'll talk about a few of these. Aspergillus fumigatus, which is very common in patients who have cancer and transplantation. It's frequently found in homes and buildings, and it's considered to be an opportunistic pathogen, meaning it rarely infects healthy individuals, but it's the leading cause of invasive aspergillosis in immunocompromised patients. Now there's others: Flavus, Aspergillus terreus, Aspergillus versicolor, and Aspergillus ochraceus. Each one of these produces one or more different mycotoxins, as you can see in this list. And so they're very important to differentiate because they can cause different, or they are associated with different disease processes in the humans. The non-Aspergillus species we test for in the EMMA mold panel are Stachybotrys chartarum, which makes macrocyclic trichothecenes, Chaetomium globosum, which makes ketoglobosums, and this is a common indoor fungal contaminant of water-damaged homes. The spores of ketoglobosum are relatively large, and due to their mode of release are not easily airborne, and that's just like Stachy. Stachy is very, very sticky and very difficult to become airborne, but its mycotoxin, the trichothecenes, are not. They're easy to get into the dust and be transported around in the house. Fusarium species. These cause fumonisins and zearalenone. Now, Fusarium, for the most part, is not documented as a real pathogen for as common as aspergillosis and candidiasis, etc. However, it has been known to cause problems in keratitis There were years ago, there was a solution for contact lenses that was manufactured by a well-known company, and that had the fusarium present in it. The fusarium then got into the eye and actually destroyed the eye. So very common in certain environmental specimens, and should be checked if you have an immune compromised patient in the environment you're looking at. The last one is a new organism that has come on the scenes recently, and that's Candida auris. This is different than all of the others in that it's a yeast, and it's found in healthcare facilities, and it can be spread through contact with infected patients and equipment. It can cause bloodstream infections, wound infections, and ear infections. This organism we have validated in a new product that we are introducing called MycoDart, MycoDart, which we'll have another webinar on. And Candida auris is very resistant to many antifungals, and the government facilities as well as laboratories are very well tuned into trying to identify Candida auris. Real-Time Lab has a environmental test, as you can see here, but we also have a human test that has been validated in the bloodstream of patients, and we can detect it in less than 24 hours if we have a blood specimen come to us. So, in review, Emma tests descriptions of mycotoxin testing also, and we've talked about these in webinars before. What are the mycotoxin assays that we do? But Emma has these 15 different mycotoxins. We have a product sheet, and you can link to that product sheet and read all about that as well by just clicking through to that. The mycotoxin panel, the 15 mycotoxins, we have 4 aflatoxins, and I've pointed out that B1 is the most carcinogenic listed by CDC and the World Health Organization. They have a gamut of problems that they can cause to patients. Ochratoxin A interferes with certain parts of the cell cycles, especially in kidney and bladder. And gliotoxin attacks the intracellular function of the immune system. So when we get a aspergillosis patient, we should be very concerned about if gliotoxin is being produced, because its immune system is being affected by the gliotoxin. So the 9 macrocyclic trichothecene that are produced are all very, very similar, but they're very different in what they do and the symptoms that they cause. And then it's associated with a disease state, as I pointed out before. So this you can read at your leisure, and this again is present in our website as to the mycotoxin panel. This also goes out on every environmental sample result that we get, and so it explains to the patients what this means. So the Emma reported that we get in a clinical specimen is now reported in fungal load. If you remember, or if you've ever seen an ERMI result, or in discussions of Hurts Me Too, they talk about the fungi being produced or being documented as spores per milligram of dust or spores per milliliter of fluid. We have adopted the word fungal load units, which are reported in nanograms per mil or micrograms per mil of fungal DNA per mil. The fungal load is a term that Real-Time Lab now will use when discussing fungal infestation and possible infection in the patient and his or her environment. So why the term fungal load? I use this, and the real-time lab decided to introduce this because the term viral load is used in HIV and some other viral diseases and viral states. So just in summary, without treatment in the body, a virus or a fungus or bacteria can replicate or make copies of itself, increasing the amount of fungus or virus in the body unchecked. Now, when a fungus grows, it usually grows and it can make mycotoxins. It can make them in different times and different amounts. The results have been reported in the past in spores per milligram of dust or spores per milliliter of fluid. Real-time lab has validated and associated these with human illnesses, but that's been very difficult to interpret because of these spores per milligram and all the environmental laboratories have the same issues. So we introduced the term fungal load, and it's a measurement of how much fungal DNA is present in a sample. And we have written a letter to describe what a fungal load is, and you can see this at the bottom. You can click through to our explanation of mold and mycotoxin assays. It's a two and a half page summary. No, it's a 2-page summary of our how we do this test. So again, fungal load is determined at limit of detection, or LOD. All fungal load tests have a cutoff point below which they cannot reliably detect fungi, and that's called LOD, or limit of detection. Just because the level of mycotoxin in our fungus, though, is too low to be measured, it doesn't mean that they've disappeared completely. So it is imperative that the patient and the inspector be vigilant in their pursuit of ridding the body and the environment that they live in of all the molds and mycotoxins that are causing a problem. And here down below is our letter that you can click through to read about fungal load. Here's the paper that we will put out with every result. This is a correlation showing the 10 organisms that we test for and what the results are in nanograms per microliter or milliliter of fluid. So let's just take one example right now, Aspergillus fumigatus. If you look at the top, it says 1 nanogram of detected DNA per mil of Aspergillus fumigatus is an average equivalent to the following number of spores per mil. Now this we've done with validations. We've received organisms from the University of Texas in San Antonio, as well as from the American Type Culture Collection. We grew these organisms, determined the number of spores by counting them on under a microscope, and then we break them up and we measure the amount of DNA in those spores. And we determined that 1 nanogram of DNA is equivalent per sample to the number of spores per milliliter of Aspergillus fumigatus. Now that takes care of 9 of the organisms that we test for. But the 10th one is a yeast called Candida auris, which is something you may or may not have heard of recently in the newspaper or on the news about a very superbug that is coming out that's very resistant to antifungals, and it can be found on patients and on the nurses or doctors' hands, hospital staff's hands. So it's a environmental sample or environmental organism. So, however, yeast grows just like every other, the Candida auris grows just like any other yeast. in that it grows in colony-forming units, not in spores, because there are no spores per se. So we have determined through our validations that 1 nanogram of detected DNA per mil of sample is approximately equivalent to the following number of colonies or colony-forming units per mil, and that comes to 0.0023. Now, if you click through to our whole discussion, if this seems a little bit complicated for you to understand, we explain this even more in greater detail, and we have a number of endnotes on it as well. So, and you're welcome to call us at any time to discuss that. So the mycotoxin reports, as we've talked about before, are reported in parts per billion or nanograms per deciliter. Each mycotoxin group has been validated to be reported in parts per billion. This is well accepted in the peer-reviewed literature and in USDA or FDA policies. So how do we collect these specimens and how do we submit them? So the instructions are part of the kit that you will receive from Real-Time Lab when you order them. We talk about suggested environmental sampling for molds or fungi and mycotoxins. The biggest note that we want to add to this is the individual taking the specimen or specimens should wear a pair of latex gloves, and that's not provided in our kit. So you have to go to a drugstore or to a pharmacy and buy some latex gloves. Pretty cheap, less than a dollar or two, to prevent any contamination that could be coming off your skin, because you may be a carrier to Candida auris as well. You don't want that to be showing up in your house unless it is there. Sampling of the environmental samples. This is a big change to what we have suggested. Any visible mold should be sampled first, and we provide a swab or a gauze, and a sterile gauze provided in the kit. Any areas of suspected leaks or evidence of water damage, either present or past, should be considered for sampling as well. Now, if you don't have any problems or anything that you can see in numbers 1 and 2 from above, then you have to use these following procedures, and this is in our sheet as well. You can take dust from air conditioner filters. This is preferred, and we take about 1 to 3 grams, and that people have asked, well, how much is that? It's a thimbleful. Samples should be placed in a biohazard bag provided in the kit or a clean baggie. We can take dust from vents, approximately 1 to 3 grams, again a thimbleful, using a cotton swab, which should be dampened with tap water, then rolled over the vents or a sterile 3 by 3 inch sterile gauze supplied in the kit as well, and they are then placed in a biohazard bag or a clean baggie. Now I can just picture people calling me saying, well, how do I use this gauze? Well, you have gloves on, so you take the gauze out of the sterile package and you just wipe it much like you're dusting your furniture at home. So the 3 by 3 gauze is how big is that? People have called and said, well, how big is a 3 by 3 inch gauze? Without measuring it, we're going to say it's the size, half the size of a normal credit card. And you can use this or you can use something of a furnace filter that's that same size, or an air conditioner filter, and then you place those in a biohazard bag. Now, you can also do a suspected portion of drywall. The problem with drywall is that the fungi love to eat the cellulose, and they like to eat gypsum. And bacteria will live in gypsum as well. That's the plaster of Paris that you all were familiar with as kids. And they don't like paint, though. Paint will absolutely inhibit our DNA test. So if you're going to send us a piece of drywall. Make sure it doesn't have the paint on it, because we will remove the paint anyway if you have it on there. Other suggestions are vacuum dust and dust on the top of shelves, under refrigerators, and areas not cleaned very often. I know some of you say I clean everything all the time. The sampling of our environmental samples, we allow a composite testing. Well, what does that mean? At your request, we can combine up to 3 samples and run them as a composite. So if you send 1 in, costs the same. 2 in, if you run them together, costs the same as 1. 3 costs the same as 1 if you put them as a composite test. You must indicate this clearly when you're ordering, if it's going to be a single or composite test, and this is on the chain of custody, which we'll talk about in a minute. So the sampling of environmental samples also is very concerning how you ship it. Each sample must be accompanied by a chain of custody form, so we're going to talk about that next, and a credit card authorization. Oh, most important. No, not most important, but important. And you must understand that no testing will be initiated without the chain of custody and or the payment. Place the biohazard bag or the baggie and all the paperwork in a shipping envelope that we provided and deliver to FedEx drop-off location for a free, I underline that, free 2-day shipping. Samples, if you want to be sending them overnight, you have to pay for yourself. This is our chain of custody form, and it's pretty self-explanatory. At the very top, the environmental company or the client, that's the patient. We'll fill that out. And then the project name. So if you have a house and you live on Hill Street or on whatever street you want to live on, you must have a project name. So you can call it Hill House or something, and give the address and who collected them, and then who shipped them, and the date that they shipped them, and what courier did you use, FedEx, UPS, or USPS. And if you do have a tracking number, that would help us as well, but we will put that tracking number down on our bottom, on the bottom sheet when you see that. But it helps us when you call us. Now the test selection is very important. You'll see EMA, which has both mycotoxin and DNA, or you can select mycotoxin only or DNA only. And you give it sample number like A or B or C, and then sample description. You have to describe it. Use your terms. And then the most important, are you combining these as a composite? Yes or no? And then we've listed that EMA tests for 10 toxigenic fungi and 15 mycotoxins. The turnaround time is 7 to 10 days. We usually try and get them out faster. And we will send them by email or by regular mail. And that should be it on the chain of custody. The final thing you must remember is keep a copy of this. Take a picture of it with your phone or do something with it, because when you call us, we will want to know what's the tracking or when did you take it or something, so we can find it as well. Turnaround time for environmental specimens is, so here again is our little mold critter traveling through the webinars as well. You can read about us on realtimelab.com, Facebook, and Twitter as well. And you can see my email as dhooper@realtimelab.com. Thank you for listening, and we look forward to doing more webinars with you in the future.